Hello, everyone, and welcome to today's live webinar, Strategies for Successful Cross-Linking and Bioconjugation Applications. The presenter is Greg Hermanson, Chief Technology Officer and Principal at Aurora Microarray Solutions, Incorporated, and President of Greg T. Hermanson, Incorporated, a bioscience consulting company. I'm Judy O'Rourke, and I'll be your moderator for this educational webcast, presented by LabRoots and sponsored by Thermo Fisher Scientific. Before we begin, I would like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please note that you'll be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window or use the Q&A button to let us know that you're having a problem. Please join me in welcoming Greg Hermanson. Greg has more than 3 decades of experience in the development of life science research and diagnostic products. He has particular expertise in protein chemistry, assay development, immobilization, and bioconjugation. Greg is well known as the author of Bioconjugate Techniques, now in its 3rd edition, which is an extensive manual on the methods of bioconjugation. He has had significant impact on the bioscience fields, as proven by more than 27,000 citations to his publications and patents. I will now turn the presentation over to him.
Thank you so much for that introduction, and thank you for everyone who is attending today. We've got a lot of material to cover, so let's dive right in. Where we're going today is, the outline is a basic introduction to bioconjugation, which will be brief, but to get everybody on the same page. And then we're going to look at 2 major areas of bioconjugation applications, namely antibody conjugation and antibody immobilization. And then finally, we'll wrap up with a section on some exciting new reagents for bioconjugation related to mass spec proteomics. Some important knowledge gained today will be some of the sites of conjugation using antibodies, functional group accessibility, forming the ideal antibody conjugate, biocompatible conjugation techniques, how to optimize antibody conjugates, and how to study protein interactions using mass spectrometry. First, let's look at what bioconjugation is. What is it in its most basic sense? It's the covalent linkage of 2 or more molecules together. Sounds pretty simple. And if you look at the diagram, molecule A and molecule B are separate, and in some way they're mixed with a conjugation agent to form a covalent AB conjugate. And the primary benefit of doing this is that the final conjugate takes upon the properties and activities of the individual components used to form the complex. Therefore, the result is a new hybrid molecule. And the process creates novel complexes with unique characteristics that wouldn't otherwise be present. And this is useful for research, diagnostics, and even therapeutic applications. Some potential reagent designs are quite extensive. Perhaps the 2 most common are right up in the upper middle. Homobifunctional compounds have the same reactive group on either end and are separated with a small spacer arm. Heterobifunctional compounds have different reactive groups on either end. So the reactive groups can be 1 or more. They can be the same or different. They can be thermoreactive, which means that they are spontaneously reactive under the conditions of the reaction. They can be photoreactive, which means they need to be exposed to a wavelength of light, usually in the UV range, to get them to actually couple. Or they can be bioorthogonal, which means that they won't couple with biological molecules, but the bioorthogonal pairs actually couple together. And they can have many different spacer arms or cross bridges present. 1 or more spacers or linkers, linear or branch systems, hydrophobic or hydrophilic cross bridges. They can even be cleavable or non-cleavable. The overall designs can be simply bifunctional, meaning two functional reactive groups, trifunctional, multifunctional, and you can even design systems with particles, surfaces, or synthetic or biological constructs. Some of the more extensive designs are on the right-hand side. In the upper panel, we can see a streptavidin molecule coupled covalently to a particle, and then a non-covalent interaction between biotin and streptavidin with the biotinylated antibody. And this type of final bioconjugate is used quite often in assay systems. Some of the terminology that we're going to use today, and you'll see quite often in the field of bioconjugation, includes a functional group, which is simply the part of the molecule that is able to react with a reactive group and form a bond. So in the right-hand side, you'll see at the top, there's a protein molecule containing amines, and it's the functional group that's going to react with a reactive group. And the reactive group is a part of the bioconjugate reagent that is able to spontaneously react with a functional group and form a bond. So the reagent, being SMCC in this example, has on its left side an NHS ester, which is able to spontaneously react with the amino groups on the protein and form an amide bond. In certain cases, a catalyst might be needed, and that's simply an additive to a reaction to facilitate the bond formation. In a few instances, a system may use two reactive groups, so a secondary reactive group may be added to the solution, and it functions along with a primary reactive group to form the final bond with the functional group. There are also activation agents that are present in some reactions, and this is simply a reagent that creates a reactive group in situ from its initial reaction with a functional group. And finally, the bond is the type of linkage formed. In this case, in the example, it's an amide bond. Over the years, there's been many major types of bioconjugates that have been developed for applications, and this is certainly not a complete list by any means, but these are some of the most significant bioconjugates that are now being used. Antibody enzyme conjugates being used for ELISAs and immunoassays of all different types. Antibody drug conjugates now being developed to target and treat tumors and other diseases in vivo. Fluorescently labeled antibodies for imaging within cells and within tissues. Biotinylated antibodies that can be used in tandem with streptavidin conjugates for assays and detections, like streptavidin enzyme conjugates, fluorescently labeled streptavidin. And then there's a whole range of drugs and proteins that have been modified with polyethylene glycol, pegylated drugs and proteins. The benefits of pegylation combined with the drugs and proteins have enhanced properties in vivo. And then many different types of antibody particle conjugates used for assay or detection. And there's also immobilized affinity ligands used for assay or purification in large scale, hapten carrier conjugates for vaccines or antibody production, and fluorescently labeled nucleotides or oligos for next-gen sequencing, PCR, or many different types of hybridization assays. All of these types of bioconjugates have figured into applications that today are literally worth billions of dollars, and some of which are used in huge quantities, such as industrial catalysis and food production, where literally kilo quantities of bioconjugates feed in to produce products. But they can also be used in biosensors, in vivo imaging systems, microarrays for cancer therapy, in ELISA-based assays to study protein interactions, for diagnostic assays in FRET-based systems, for detection in Western blotting, as vaccines, in affinity separations, in fluorescence imaging, and in drugs and biologics. So you can see that bioconjugation has had major impact in many different fields and huge applications. Let's look now at antibody conjugation. Antibodies are certainly the most popular component used to create bioconjugates. Perhaps 70 to 80% of all bioconjugates use an antibody as part of their final complex. the 3D structure of an antibody molecule, an IgG type, can be seen here in this illustration. The antigen binding sites are on the outer portion of the Y, the upper Y, and there is 4 polypeptide chains that wrap around each other. The FC fragment consists of 2 heavy chains that are kind of looped around and form a donut hole in the middle. And the heavy and the light chains in the upper portion, the Fab fragments, also do something similar and kind of form a donut hole in the middle as well. The most common targets for conjugation on antibodies are amines, thiols, and glycans, or the carbohydrate sugar OH groups. But the complex 3D structure of an antibody like this can actually alter the accessibility and reactivity of some of the functional groups within it. And that's due to the folding and the relative accessibility of a functional group within it to the solvent layer during the conjugation process. And it turns out that the degree of surface exposure of a functional group on an antibody correlates very well to its relative reactivity and ability to be modified. And of those functional groups, amines are by far the most popular that are typically targeted with antibodies. In this illustration of the same antibody, I've highlighted all the lysine side chains in yellow. So you can see how many lysines are present in this actual molecular model of an antibody found in the Brookhaven database. And this one has a total of 81 lysine residues. Antibodies typically do contain dozens of amines from lysines and the N-terminal of its 4 polypeptide chains. Many of the lysines in antibodies are solvent accessible and easily modified, but some may be post-translationally blocked, such as the N-terminals are often acetylated, or not sufficiently accessible on the surface to allow a coupling reaction to occur. If we look at the next slide, I've blown up the FC region of that antibody. And you can see that some of the lysine side chains can be seen to actually stick out from the surface of the antibody molecule. Others, however, are less accessible or almost completely hidden within the 3D structure. It turns out that this degree of accessibility correlates to the degree of reactivity with amine reactive groups in bioconjugation reagents. Every protein will, to a degree, behave this way. There will be accessibility of some residues and inaccessibility of others. Other factors that affect coupling include whether the lysine amine is involved in salt bridges or hydrogen bonding, which both decrease reactivity. This can be seen if you try to modify an antibody molecule with a small hapten, such as with the thiolation reagent, SATA or SATA. And if you look at the reaction efficiency and yield of its amine modification, you'll learn something interesting about the relative accessibility of antibodies. So SATA contains an NHS ester on one end of the molecule, as can be seen in the left-hand structure, and a protected thiol group on the other end. The NHS ester reacts with amines to form an amide bond. The protected thiol can be deprotected with hydroxylamine, and then the thiol is actually measured using Elman's reagent. So from the graph, you can see that the efficiency of modification can be seen to be very good at low levels of SATA addition. In other words, for the amount of SATA added, you get a fairly good yield of the number of amines actually modified. So there's a population of amines with high activity. However, as the amount of SATA is increased, you'll notice that the resultant level of incorporation plateaus and levels off. And that's due to there's a population of amines present on every antibody molecule that are easily accessible, some that are intermediate accessible and reactive, and then others that are simply not going to be able to be modified if the antibody is in its native state. The other major target on antibodies are thiols. And thiols, too, have an accessibility issue. Thiols are the second most targeted functional group in antibodies. However, there are no free thiols available in IgG molecules, only disulfides. And they exist, if you look at the structure, molecular model, the polypeptide chain is shown as a ribbon structure, and the cysteine disulfides are shown as space filling within the structure. So they exist between the heavy and light chains. And if I can just show you where that region is, it's right in that area there. And there's one disulfide holding each of the light chains to the heavy chains in that region. And they exist between the heavy chains also within the hinge region, and that's right in this region below the heavy light chain interaction. And they also exist within the globular regions within each subunit. Now, fortunately, with antibodies, the disulfides can be reduced under non-denaturing conditions to create free thiols. The caveat is don't use high concentrations of reducing agent in order to avoid breaking down the total antibody structure. It has been determined that about 3 to 5 millimolar DTT or 2-mercaptoethylamine or TSEP is probably the best range to be in, because at that level you can create 4 to 6 thiols for conjugation without tearing apart the subunit structure of the antibody molecule. The third most targeted group within antibodies are the glycans, or the carbohydrates. Now, glycans and IgG are typically attached in the Fc region, but unlike the stick structures that you sometimes see where they show the carbohydrate actually sticking off from the heavy chains, they're actually found between the heavy chains. In this molecular model on the right, I've highlighted the carbohydrate in yellow, and the rest of the molecule is shown space-filling, colored according to the atom type. So you can see that the yellow is almost entirely between the heavy chains, in between the pocket or the donut hole formed from the two heavy chains wrapping around each other. So glycans are nearly hidden within a pocket with limited accessibility. Now, there is an advantage to targeting glycans, and that's to avoid blocking the antigen binding sites. The same with thiols. But the tip in this case is to use longer bioconjugation reagents in order to reach the functional groups within the glycan structure. And one way of targeting glycans within proteins and IgG molecules is to oxidize them with periodate to create aldehydes for conjugation. And in the lower left-hand structure, I've circled 2 beta-D galactose residues which have diols on them, and periodate will cleave the carbon-carbon bond between the diols and oxidize the hydroxyls to aldehydes. And aldehydes can be conveniently coupled with any hydrazide or amino-oxy-containing reagents to form hydrazone or oxime bonds, respectively. So people often ask, what is the ideal antibody conjugate? And you may think that an ideal antibody conjugate might be an equal mole ratio of components in the final complex. In other words, a 1-to-1 ratio. In the upper equation or reaction scheme, an antibody is seen to be reacted with an enzyme in the presence of a crosslinker, and you get a 1-to-1 complex of antibody to enzyme. And that ideal complex 1-to-1 is something that is sometimes thought of as being the most ideal. However, you may also think that having an exact ratio of one molecule attached to another molecule, for example, 4 enzymes linked to each antibody, might be the ideal. So you can react an antibody with 4 enzyme molecules in the presence of a crosslinker and end up with an antibody with 4 enzymes attached. Unfortunately, forming these types of the ideal conjugates is difficult or almost impossible to achieve. And the reason is something called the conjugate distribution. And this is lessons from the hapten labeling of BSA, a paper published in 1996 in Bioconjugate Chemistry, looked at the coupling of 5 hapten molecules, NHS fluorescein, NHS coumarin, danzyl chloride, an NHS succinate derivative of digoxagenin, and NHS biotin at the bottom. And the reaction was done on bovine serum albumin, which has 60 amino groups. And each of these haptens were reacted in molar ratios of 5-to-1, 15-to-1, and 30-to-1. in phosphate buffer at pH 8, in a buffer DMF mixture, 4 hours at room temperature, and a certain number of those amines will become modified with each of these haptens. And what was found was that at each level of molar excess for these haptens, we're looking here at the results of NHS biotins specifically, a distribution of labeling yields was observed, even exceeding the mole ratio for a small portion of the modified BSA. The results indicate that bioconjugates end up being a mixture of different mole ratios in the final complex. So if you look at the 15-to-1 mole ratio in the middle, you can see that there's many populations of numbers of biotins on each BSA molecule. And there's an average somewhere in there that may be 12 modifications. But the distribution will always be present in every bioconjugation complex that you can form. So some bioconjugation facts of life to realize is that with regard to conjugation, modification, or even immobilization reactions, the reactions are never 100% efficient. Hydrolysis and other competing reactions will decrease the yield. Functional group accessibility affects the efficiency of reactions. And reactions done with amines on proteins, because there's many of them, often give modification levels that consist of broad distributions. And the realities of trying to achieve the ideal antibody conjugate using a certain molar ratio in a reaction won't yield the corresponding exact ratio of linked components in the final complex. And also consider this, using a low mole ratio to form an antibody conjugate may actually result in a significant portion of the antibodies with very little modification or no modifications at all. So if you try to biotinylate an antibody and achieve a 1 biotin per antibody or a 2 biotin per antibody, you will always end up with some antibodies with no biotin at all on them because it will always be a distribution. Now what about antibody conjugation? There's 2 types of reagents that are probably used most often when you're looking at antibodies and proteins coupled together, like antibodies and enzymes. Homobifunctional reagents and heterobifunctional compounds. Homobifunctional reagents are the oldest crosslinkers available. They date back probably to the 1950s, maybe even to the 1940s, in the use of formaldehyde to make protein crosslinks and glutaraldehyde after that. And then some further developments in these types of reagents can be seen with BS cubed, having 2 sulfo-NHS esters at either end, making the compound itself water-soluble, and with DTPP with a cleavable disulfide in the middle. Heterobifunctional reagents were designed to try to improve the conjugation process over homobifunctional compounds, and some of the most popular are sulfo-SMCC and SMCC at the top of the second column. Sulfo-SMCC has a water-soluble sulfo-NHS group, which solubilizes the entire compound better than the aliphatic NHS ester would. And then at the bottom, NHS PEG-12 maleimide is a water-soluble, very hydrophilic, similar crosslinker to SMCC, but it has a PEG spacer arm which provides water solubility to the entire compound. Now, when using homobifunctional reagents to make antibody conjugates, remember homobifunctional reagents react with the same functional group on both ends. So cross-linking antibodies through amines will result in multiple linkage sites on each protein. Conjugates, therefore, typically end up being oligomers, and they may even precipitate from solution. The result is that a broad distribution of species will form, and some of them will be so large that you'll lose some of the yield due to precipitation. And scaling up the reaction is very difficult because you can't control the reaction well. The main example of this type of conjugation is the use of glutaraldehyde to make antibody enzyme conjugates. Antibody conjugation using heterobifunctional reagents improves on the homobifunctional conjugation scheme because they have 2 different reactive groups, 1 at each end. So they're used in a 2-step process. In the first step, the antibody molecule is modified through one end of the crosslinker. that's able to react with, say, the amines on the antibody molecule. That forms an intermediate modified antibody containing reactive groups sticking off from the antibody, which is purified away from excess crosslinker. Then the second molecule, in this case an enzyme, is added in a mole excess, and the final enzyme antibody conjugate is formed. Still a distribution or population of different modification levels, but much more controllable in that you don't usually get oligomers or precipitation problems in the final complex. So how do you make better antibody conjugates? Some choices for bioconjugate design might be better than others. Bioconjugation methods that use, for instance, the older aliphatic or hydrophobic linkers to couple biomolecules may actually cause problems. So the original older crosslinkers and modification reagents were all hydrophobic. The disadvantages of using hydrophobic linkers include a strong potential for aggregation, even when using heterobifunctional and two-step crosslinking methods, nonspecific interactions with linker arms, off-target effects, or high background in assays. Newer options, fortunately, now are available for incorporating more biocompatible linkers into conjugates, using conjugation reagents containing polar groups or hydrophilic linker arms, and using polymers that can add hydrophilicity and biocompatibility. So biocompatibility actually can be designed into reagents and conjugates by modifying or coating surfaces with hydrophilic reagents before conjugation. You can modify particles with hydrophilic reagents before immobilization. You can use hydrophilic crosslinkers instead of aliphatic or hydrophobic ones. There are even hydrophilic biotinylation reagents and modification reagents available. So you can help to avoid the precipitation or aggregation problem by choosing hydrophilicity over hydrophobicity. So examples of hydrophilic or biocompatible molecules that have been used are Hess, agarose, dextran, PEG or polyethylene glycol, and PLGA or polylactate glycolate. And these have been used to stabilize biomolecules as multifunctional scaffolds, as water-soluble spacer arms in reagents, to increase drug half-life in vivo, to enhance pharmacokinetics in therapeutic conjugates, and to eliminate nonspecific interactions in a final bioconjugate. Now, if we focus on discrete PEG compounds in particular, and these are discrete molecules versus the older polymeric PEG polymers that have been used for decades now to modify some drug candidates to make them water soluble. Normal polymers of ethylene glycol are polydisperse, and typically they have large molecular weights ranging from 1 to 40 kilodaltons. On the right-hand side, you can see an analysis by capillary electrophoresis of a 5 kilodalton PEG polymer, and it in reality has dozens and dozens of species within that preparation of different chain lengths. And even if it's reacted in the lower graph with a 20-base DNA oligo, the result is even worse distribution of molecular weights because the initial PEG polymer was so polydispersed. By contrast, discrete PEG reagents consist of a single molecular species having a known number of ethylene glycol repeats. So you can actually choose now hydrophobic or hydrophilic reagents depending upon the application. And in some cases, it's still appropriate to use hydrophobic reagents, especially if a conjugate has to penetrate cell membranes or the use of it has to be in a hydrophobic environment. Discrete PEG-based analogs of traditional cross-linking or modification reagents can provide increased water solubility, decreased stickiness, better accessibility, and no charge. So in the biotinylation reagents on the lower left-hand side, the most popular biotinylation reagent still is probably NHS-LC-biotin or sulfo-NHS-LC-biotin. The alternative PEG-based compound might be NHS-PEG-4-biotin, with the PEG chain providing hydrophilicity and water solubility. And there's even longer chains available. As modification agents, we looked at SATA reaction with antibody molecules, You can choose a hydrophilic SATA-like compound, NHS PEG-4 thioacetate, to do exactly the same thing, but the modifications will be hydrophilic instead of hydrophobic. And one of the more popular heterobifunctional crosslinkers, SMCC, is hydrophobic and water insoluble, whereas NHS PEG-4 maleimide is hydrophilic and very water soluble. And this slide just illustrates the difference between using sulfo-NHS LC-biotin. Now, sulfo-NHS group is negatively charged and will provide initial water solubility, but the rest of the molecule is very aliphatic and water insoluble. So if you modify an antibody molecule with sulfo-NHS LC-biotin and compare it to NHS PEG-12 biotin, you see that the result in the hydrophobic case of NHS LC-biotin precipitates some of the protein. In the hydrophilic case, it does not precipitate. In fact, it increases the solubility of antibody molecules. So some of the advantages of using discrete PEG-based reagents for antibody conjugation over aliphatic ones, or the older type reagents: you can decrease aggregation and precipitation, increase the stability, increase specific interactions because you don't have as much off-target effect, increase signals in assays and detection systems, decrease nonspecific binding and background, and therefore increase signal to noise. They actually stabilize proteins and other molecules. They are very flexible with increased freedom of motion, and you're able to use higher modification levels in conjugates to increase activity. That's one thing that's being found right now in the manufacture of antibody drug conjugates. If the drug is immobilized on the antibody using a hydrophilic PEG linker, they can get more drugs on each antibody without precipitating the complex. Now, there's one caveat with the use of cross-linking reagents or modification reagents with protein conjugation, and that is every time you use them, you will almost always end up having some dead ends, and they can come back to haunt you. These are leftover modifications that never got coupled with another protein or molecule. And dead ends can actually change the surface properties of antibody conjugates, even if they've been blocked so that they're no longer reactive. Antibodies that initially might have a tendency to aggregate may aggregate even more with some dead ends, especially if the dead ends come from hydrophobic linkers and crosslinkers or modification agents. Hydrophobic dead ends can cause decreased antibody and protein solubility, increased tendency to aggregate, decreased stability of the conjugate, increased nonspecific binding, and off-target effects. Choosing a hydrophilic compound, the dead ends can provide increased antibody and conjugate solubility because the reagent itself modifying the protein or antibody is hydrophilic, decreased tendency to aggregate, increase conjugate stability, and decrease nonspecific binding and off-target effects. So how do you create the optimal antibody conjugate using everything, all the knowledge that you may have? And if you follow this process, I think you'll end up with an optimal performing conjugate for the application. So first, design the conjugate with the final application in mind. In other words, the best performance test during the development process is the actual use of the conjugate in the desired application. Then look to the literature for starting points on the reagents, reactions, and conditions for use. Use hydrophilic bioconjugation reagents when possible and if appropriate. Optimize the conjugation method to achieve the highest possible activity for the conjugate, and that means testing different concentrations and molar ratios of the components in the reaction, different buffers and additives, different pH of reactions, different temperatures, etc. Be careful not to include substances that might interfere with any of the reactions. In other words, tris is often present in commercial antibodies, or imidazole is added sometimes to buffer. Those are problematic in amine reactions. Use care to prevent exposure to conditions that might damage the conjugate or its components. For instance, strong light, oxidants, reducing agents, azide. and use design of experiment methods, DOE methods, to test multiple factors simultaneously during the development process to get to the ultimate or the optimal manufacturing methods to create the best conjugate possible. Also be certain to optimize storage conditions to ensure long-term stability and make several batches using the final process to ensure conjugate reproducibility. It can be made again and again. Now let's look a little bit at immobilization of antibodies onto particles and surfaces. This is a large application area for antibodies today. But there's also tremendous diversity of particle compositions. You can have many different forms of organic polymers, from polystyrene, very hydrophobic, to more hydrophilic, hydroxyethyl methacrylate, and some of the newer polymers containing cycloolefin polymers. You can also have inorganic particles consisting of silica, gold, silver, and many different types of semiconductor alloys for quantum dots. You might also have composites and advanced polymer constructs. Perhaps one of the most frequently used particles is superparamagnetic for use in assay systems. There's also biodegradable particles, liposomal particles, and many different types of dendrimers and dendrons that can be used to immobilize or couple antibodies to. And there's a wide range of particle sizes. The sizes can be particulate, 100 micron size or greater, down to 10 micron, or probably the most popular range is from 500 nanometers to 2 microns right now for immobilizing antibodies for use in assay systems. And most diagnostic companies that use particle-based assays use that range of particle sizes in superparamagnetic particles. But they can be down even as low as molecular size, down to 10 nanometers. And if you look at that little spot in the 10 nanometer particle size, these are drawn to scale, that one can still couple molecules like antibodies and other proteins in good yield because the particle is on the same order of magnitude and size as the antibody molecule. There's a few things to keep in mind with particles, however, and there's physical and chemical considerations that are unique to particles that you don't run into when you're coupling with other proteins. Namely, as particle size decreases, if you think of each particle as a molecule in solution, the molar concentration of particles in a constant volume of solution increases. That's for a given mass of particles. Also, the ratio of its surface area to mass increases. So as the particle decreases in size, you get more surface area, you get a greater number of potential reactive groups present too, which can lead to greater reactivity. There's also a concept with commercial particles called parking area, and that's the average area on the particle surface that is occupied by a functional or a reactive group. And depending upon the manufacturer of the particles, it can vary widely. Aldehyde modified particles, if you look at it in the literature and what's available commercially, the typical density range of particles is 100 to 230 square angstroms per aldehyde group. And for carboxylated particles, the density range is typically somewhere around 10 to 125 square angstroms per carboxylate group. Depending upon the density, you may have more or less surface exposed on the particle surface. And if the surface is hydrophobic, it may be detrimental to the overall performance of the particle. So hydrophobic or hydrophilic surface characteristics can come into play. They affect protein stability and activity and affect the particle aggregation and nonspecific binding potential. But fortunately, just like with bioconjugation reagents or crosslinkers and modifiers, you can design biocompatible particles in much the same way. Some of the strategies for creating biocompatible particle surfaces include the use of hydrophilic polymers or a polyhydroxylic layer. Some people have even used multiple protein layers to mask the protein particle surface, or use hydrophilic linker arms to separate affinity ligands from the particle surface. particle surfaces in general that have abundant hydrogen bonding sites with water molecules, in other words, they're hydrophilic and hydrogen bonding, they seem to perform the best for biomolecule targeting with antibodies coupled to them. And that includes particles made of p-hema, polyvinyl alcohol, other hydroxylic particles, agarose-coated ones or carbohydrate-based surface coatings, and PEG or PEG polymer networks. Turns out that hydrophilic particles decrease particle interactions, in other words, aggregation, and decrease nonspecific binding to biomolecules. This has been done on surfaces with PEG-based linkers, and Prime and Whiteside published in 1991 a SAM surface on gold layers consisting of different length PEG chains. And some of the PEG chains terminated in OH groups and were not reactive or functional in typical coupling schemes, while other longer ones terminated in carboxylic acid groups, which could be used for coupling. And what they found was this type of layer performed beneficial in terms of creating this biocompatible surface and low nonspecific binding character. This type of surface is sometimes called flowers in the grass. You can do the same thing with particles. Many particles are available as carboxylate derivatized particles. If you take a particle, even one that has carboxylate groups and the particle itself is hydrophobic, you can couple to that an amino PEG and carboxylate modifying agent and use EDC sulfo-NHS reaction and a two-step reaction to couple the amine end of the spacer arm to the carboxylate group on the particle, forming a long chain PEG particle terminating in carboxylates for further bioconjugation. And the result of that is the hydrophilic PEG particle provides the hydrophilicity to reduce the nonspecific binding and aggregation that is seen so often with particles. And the same flowers in the grass approach can be used with particles containing carboxylates, so you get a nice coating of pegylated molecules on the surface of a particle which totally block the nonspecific binding character, yet you still have functional groups sticking off that provide conjugation capability for antibodies. And they can be coupled, antibodies or other proteins, to these carboxylated modified particles using a carbodiamide reaction like EDC. Now, EDC is probably the most popular conjugating agent to particles. And it's used as a zero-length crosslinker that directly links a carboxylate group with an amino group on an antibody or another protein molecule. And this is a typical reaction scheme where EDC reacts with a carboxylate, forming an intermediate ester that then reacts with the amine to form the amide bond. However, EDC does have some potential deficiencies. And I'm not going to go into detail in the complete reaction scheme of EDC other than to point out that there's 2 points in this reaction, complex reaction scheme, where EDC can be lost to hydrolysis. There's also 2 points where EDC intermediates can be lost to rearrangements, both of which can decrease the yield of the reaction considerably. The desired product, of course, is to react with the amine and form the amide bond. But you can see just from this reaction scheme, there's a lot of yield loss potential in a typical EDC reaction. EDC reactions can be made more efficient, however, by limiting some of these side reactions through an initial reaction with NHS or sulfo-NHS. This is a two-stage reaction that creates an intermediate sulfo-NHS or NHS ester on the particle. And the intermediate has benefit to particle stability because the negative charge on the sulfonate of the sulfo-NHS will actually cause particle repulsion and decrease aggregation capability in the intermediate activated form. But since that sulfo-NHS ester hydrolyzes slower than the EDC ester, and since it's not subject to the same rearrangement products as EDC esters are, you get a much better yield of the amide bond formation if an EDC sulfo-NHS ester reaction is used to replace the standard EDC reaction. Now let's look briefly at bioconjugation for mass spectrometry. There's a new trend to use cross-linking reagents in the study of protein interactions and other protein-related research, and couple it with mass spectrometry to determine exactly what proteins are being cross-linked and where they're being cross-linked. So using this method, you can study protein structure, in other words, elucidate and confirm 3D protein structure. You can even identify conformational changes as proteins bind to other molecules or are activated or deactivated. You can demonstrate the peptide folding within a protein molecule. You can study very complex multi-protein complexes through cross-linking with these types of reagents. And you can study protein interactions. In other words, cross-link interacting proteins to capture them and then use mass spectrometry to determine what proteins were ultimately cross-linked. One such reagent that's becoming popular is disuccinamidyl sulfoxide, DSSO. It's a new specialty crosslinker designed to capture and identify protein interactions using mass spectrometry as the readout. It links peptides between amine groups, and it's cleavable in second stage MS. And this just shows the synthesis of the molecule, which is made from a thiodipropionic acid precursor and then forming the bis-NHS ester intermediate and then oxidizing the thiol in the middle to the sulfoxide. This crosslinker, homobifunctional and cleavable in MS2, can be reacted with two proteins that are interacting through their amine groups and forms two amide bonds between the two proteins. So now the two proteins have been cross-linked and captured, and if they were interacting proteins, mass spectrometry can determine exactly what the interacting proteins were. These are then subjected to proteolytic digestion to create small peptide fragments that are cross-linked, and then in MS2, one side or the other of the sulfoxide is cleaved, and these cleavage peptide products are then read out in the mass spectrum, and software is used to determine what peptides were cross-linked and what proteins they came from. In a workflow, over on the left-hand side, sample preparation can consist of cells grown in culture, treated somehow to study a particular protein interaction. DSSO is added and cross-linked all the proteins within the sample. The cells are lysed and sonicated. The proteins are denatured and digested and then separated in size exclusion chromatography and then injected into an Orbitrap fusion Lumos system, which is designed to handle this type of crosslinker and the readout from it. I won't go into great detail on the software, but the data analysis facilitates the determination of exactly what proteins were being cross-linked in the sample. So MS cleavable crosslinkers actually can revolutionize the study of protein interactions. Previously, before the capabilities of mass spectrometry, identifying what proteins were interacting after cross-linking with homobifunctional compounds was extremely difficult. Now, the rapid analysis of DSSO crosslinks by mass spec can identify the exact proteins that were cross-linked. So here we have peptide A cross-linked with peptide B. After MS squared analysis, we get peaks representing peptide A linked to the cleaved linker, and also peptide B in the mass spectrum shows up linked to the cleaved linker, and then the software handles the interpretation or deconvolution of the data. Just a few things I want to mention in conclusion. I want to thank Thermo Scientific for sponsoring this symposium, and there's two important things that I think you should consider in your cross-linking needs, and that is the Thermo Scientific Pierce premium grade reagents, which are a higher level of high purity reagents that can be used to create all kinds of derivatives, labeled proteins, and bioconjugates. They undergo enhanced testing, lot retention, and all the controls necessary for critical manufacturing to be done. And also a very convenient product, Thermo Scientific no-lay reagents. And this allows either bioconjugation or modification reagents to be dried within a strip well or within individual containers in ready-to-use amounts. Once reconstituted, the reagent is ready to use at the desired concentration, and it helps reduce the problem of weighing out an amount of crosslinker or modification reagent to use in your application. It's very convenient. And finally, for additional information, I want to direct you to Bioconjugate Techniques, the third edition of my book. It's in full color with a lot of proven methods and protocols for synthesizing all different types of bioconjugates. You can find step-by-step presentation of protocols, full color illustrations, and the new edition features about a 120-page introduction to the vast field of bioconjugation and one of the most thorough overviews of immobilization chemistry ever presented. And I'll conclude with some notes, disclosures from Thermo Fisher. For more information, you can go to my website, greghermanson.com, for your consulting needs, or Thermo Fisher's website as well for any of the crosslinkers or modification concepts that you may have. I want to thank you again for the opportunity to present, and I'll now turn it over again to our moderators.
Thank you, Greg, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the send button. We'll answer as many of your questions as we have time for. So let's get started. Our first question is, what is the best functional group to use on antibodies for conjugation?
Oh, that's a very good question. The three main ones, as I mentioned in the talk, are certainly amines, thiols, and if your antibody is glycosylated, the glycan portion or the carbohydrate portion of it. And amine modification is probably the simplest because amines are present on every antibody at various locations. It's rare to have an amine group modification cause antibody problems. In other words, it's rare to get the antigen binding site to be blocked, and it's rare to cause denaturation and loss of activity. So I would say the amine groups are by far the most popular and the simplest to use. Secondarily, I think to conjugate away from the antigen binding site, I would definitely target thiols by mild reduction. And if your antibody has carbohydrate, then I'd also consider that too. But some antibodies don't have carbohydrate. Recombinant ones prepared in E. coli certainly would not. Even some hybridomas, monoclonals, do not. So amines are certainly number one, thiols number two.
Your next one is, I have a biotinylated antibody that slowly precipitates in storage. How can I overcome this problem?
That's a frequent problem that I hear, and most often that is caused by the use of hydrophobic biotinylation compounds. Even when an antibody is modified at low levels of biotinylation, in other words, you only have a few biotins per antibody molecule, over time, if it's a hydrophobic reagent, it will aggregate the antibody and cause precipitation and loss of activity upon storage. And this may take weeks or months to occur, but many diagnostic companies have even found that the use of NHS-LC biotin, which is hydrophobic, can cause loss of activity and a total shelf life of only about 6 months of a biotinylated antibody. So I would definitely recommend looking at hydrophilic biotinylation compounds as an alternative. In other words, the ones that have a PEG-based spacer, and they will actually overcome the precipitation issue, and they won't aggregate the same way as the hydrophobic biotinylation compounds do.
We are almost out of time. This will have to be our last question. What is the best crosslinker to use for antibody conjugation?
That's an excellent question. As you probably know, there's dozens and dozens of crosslinker choices available, and oftentimes it's difficult to nail down the right one to use or the best one. Certainly today, even today, probably the most frequently used choice is SMCC or sulfo-SMCC. And those two crosslinkers were developed decades ago and have been used for antibody enzyme conjugation for many, many years. Research use and diagnostic use repeatedly uses those two crosslinkers. Again, though, those two are hydrophobic in that the reagent itself that gets modified on an antibody molecule will cause hydrophobic dead ends to be present. And that can have an effect upon the resultant performance of the conjugate because the dead ends, if they're hydrophobic, they can cause nonspecific binding and aggregation. So I would say that type of crosslinker, in other words, one that is amine reactive and also thiol reactive on the other end, but perhaps choosing a hydrophilic alternative to that type of crosslinker would perform better than the older aliphatic and hydrophobic ones. And I think you'll find better stability in reagents if you use hydrophilic reagents or PEG-based ones, and you'll find lower nonspecific binding and better signal to noise in whatever assay system you're using them in.
Great. I would like to once again thank Greg Hermanson for his presentation. I would also like to thank LabRoots and Thermo Scientific for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through May of 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.